All right, thanks, Sharon, and thanks everyone for joining. My name is Bill. I am the lead for the GDC User Services team, and today I'd like to talk to you about GDC frequently asked questions. And as a member of the User Services team, I do see a lot of these. So today for the agenda, first, we're going to start with an introduction to GDC documentation. In the spirit of questions asked by end users, I want to introduce where a lot of answers to these questions can be found in various places. Then the biggest part of the presentation is going to be specifically GDC FAQs that will give answers to these FAQs and possibly more in-depth explanations than you would see on the website. When that's done, we'll look in the Q&A section. So any questions you have, just type into the Q&A form in WebEx. And any questions we have from the audience, we will be happy to answer. And then after that, just for fun, we're going to do a short quiz. And if you'd like to participate, you can type the answer into the chat form in WebEx. And then after the quiz, if there's more questions and we have time, we'll be happy to answer those. So getting started, I just kind of want to show you around where the GDC documentation can be found. So this is an outline here, but we're going to start at the GDC documentation website, docs.gdc.cancer.gov. This is where the user's guides for all of the GDC features can be found. Each of these user's guides specifically shows you how these features work, and in some cases, specific commands you can use to run them, like for the API, for example. So whatever you're interested in using, you just click on that particular feature, and usually there's a bunch of subcategories there. Next, I'd like to show you the GDC website. This is a bit more higher level information, but it does have some good features about how the GDC works. So first, I'd like to show you where you can find the monthly webinars. And I know a lot of you may have clicked on the banner from the GDC, or perhaps you found this yourself, but what you would do is go to the support tab, click on monthly support webinar, and the recordings for all of the previous webinars should be there. The June one will be coming soon, but the rest of them are present. So they can be viewed here or directly on YouTube if you want to follow the link. Additionally, you can sign up for the current webinar, or the upcoming webinar here. Second, I want to show you where you can find the GDC FAQs. And this can be found at support, and go down to anything that says FAQs, it'll all lead to the same page. So GDC API FAQs, I will click on here. And all of the different categories are lined up here. If you unclick all of them, you will see all of the different frequently asked questions with answers once you click the plus. And today's presentation is going to be more or less aligned to these categories. So it's divided up into these categories. Now, I want to say we're not going to go through all of these questions. So if there's any in particular that you're interested in, feel free to type that into the Q&A section, and we will go through that. And then there's going to be some other frequently asked questions that aren't here yet because they are questions that came through the GDC help desk. And these are questions that I think would be valuable to demonstrate in a webinar. So those will likely be on this page sometime soon, but for now they are not. They're only in the webinar. So lastly, I'd like to show you how to reach the help desk. And the best way to do this is to go to the support tab once again. And the help desk button here, you can copy this email address, or you can click here and I think it'll open like Outlook or whatever you have. And it's support@nci-gdc.datacommons.io. This leads directly to my team, and we will always answer your questions, or we might not answer your questions if it's very complicated and we need to get some information, but we will give you a response within 1 business day. So if you have any questions and you can't find documentation, always feel free to contact us. So going back to the presentation here, we're going to start with some frequently asked questions, and the first section is on analyzing data. So the first question is, how can I tell which of my files comes from tumor or normal tissue? You may have a group of files that you are downloading from the portal, and it may not be obvious which tissues they come from. So what you would do is put the files into your cart, go to the cart button on the portal, click on the sample sheet button that is outlined here, and then download that. Once that file downloads, it'll download a TSV that I've opened in Excel here. And it shows you information about each file, like their ID and their name. But the most importantly, they have information like the case ID, which is, which patient or case it came from, the sample ID, and then the sample type specifically. And you'll see here that each of these files have two sample types because they are whole exome sequencing, I believe. So that would be comparing a tumor and normal. But if you had something like RNA-seq, you would only see one there. There are other ways to download the sample type too, like for example through the API. So I'd recommend watching the API webinar if you're interested in that, or contacting the help desk. Whoops, wrong button. Here we go. So the next question, let's say you are using whole exome sequencing or targeted sequencing. And as you know, the target capture kits are used to target specific regions of the genome. How can one figure out which target capture kit was used in these instances? And while you can't see this in the portal, we recommend retrieving this information from the API. The specified field is this here. It's fairly long, but it can be copied and pasted. And there are some other fields that you might come across, like target capture kit catalog number or target capture kit name. Those have mostly been deprecated in for simplicity, so that only the target capture kit name is there. If you're interested in the bed files, so a list of the regions that are specifically targeted, we recommend contacting the manufacturer. GDC may not be allowed to distribute these bed files, and we don't want to take any chances. So manufacturers, I know they'll sometimes have nice interfaces to download these files, and also I'm pretty sure you can just email them, and that should be fine. A question we do get often is, does the GDC correct for batch effects? And the answer is no. The GDC does not correct for batch effects. And that's for a few reasons. Correcting for batch effects depends on which samples are present in your group. And while the GDC does have a set group of files there, you may be using a smaller subset of that. And a smaller subset would mean less removal of anything interesting. So because we're always adding new samples too, it wouldn't be feasible to continuously do batch effect corrections as new samples are added. Certain projects will get, just 10 more cases for a data release. And some are all at once. So another thing is that batch effect correction, to do it properly, sometimes requires manual adjustments, which the GDC is much too large to do. And automated batch correction may remove true biological signal, especially when dealing with larger data sets. The GDC, we recommend that users perform their own batch effect correction. And if you want to hear more information about this from a bioinformatics specialist, I'd recommend watching the webinar from July 2020 about GDC RNA-seq data processing. He goes pretty in-depth there. So you can find that on the webinar page. And I'm going to plug a few previous webinars here, so you'll see that throughout the presentation. The next question, how is survival data generated in the GDC Exploration Portal? When you go to the GDC Exploration Portal, one of the things that will likely catch your eye is the survival plot that is um, kind of dynamically generated as you filter different samples in the exploration portal. So in order to generate this, the following fields are pulled out of the data and used to generate the estimate. So first is data on the time to a particular event, in this case, death or last follow-up. Information on whether the event, death specifically, has occurred, so alive or deceased. And then the data split into different categories or groups, like gender, for example. Um, and this information is used to with a Kaplan-Meier estimation, and that's how these values are generated in the API. So here's the equation here. I think this was pulled from Wikipedia, but in general, it's at a specific point in time, one minus the number of deaths that are known, minus uh, the number of deaths that are known over the number of patients that are known to still be alive at that time point. So this removes the people who are lost to follow-up from this equation and estimates accordingly. This information can also be pulled from the API. So if you don't like the visualization that you get from the portal, you can pull the data points directly, and you wouldn't have to do the estimation yourself. It's done through the API. So you would just have points you can plug into R or Excel or something and generate it yourself. Okay. Getting to the next question. Where does the mutation in the GDC Exploration Portal come from? Speaking of the Exploration Portal, you may go there and you'll see a lot of mutation data. Um, and where it comes from is whole exome and targeted sequencing somatic variant calls. And specifically, these are the variant calls that can be found in the open access map files in the repository part of the portal. So you can download all of the files that are used to generate the exploration data, if you are interested in that, or you can download these mutations from the exploration portal. Each of the project has either whole exome sequencing or targeted sequencing. They will never be mixed together. Um, if a project has both, only whole exome sequencing is prioritized, but target sequencing is used if no whole exome sequencing is available. And if you're interested in how the DNA sequencing is processed to a further degree, a lot of information can be found in the September 2020 webinar about DNA-seq processing. So one thing you may come across are clinical fields about the same patient that seem to contradict each other. And I'm here to tell you that this is normal. This comes up often. And one of the reasons for this is that some projects that are hosted by GDC are still in progress. Updates may actively be happening. New data releases may be new clinical data. And what happens is clinical data in GDC is available in two major forms. One are static files that are on the data portal, and other ones are the metadata that can be pulled from the API. And because static files are static, they can't just be changed very quickly. They may fall out of sync with what's in the API. One of the specific reasons outside of this that you might see some discrepancies in clinical data is that time point values are sometimes calculated to a month's precision and other are calculated to a specific day. If you compare these values, you'll sometimes get results that can result in differing or unusual numbers. So here's an example here, but I'll put the same thing on the next page. This is not real data, by the way. I just use October to demonstrate that it's a specific month. But let's say a patient had a follow-up appointment in October, And that's all we know. The doctor wrote down October. But the patient died on October 13th. So because we only know the patient had a follow-up appointment in October, it may be automatically estimated to be October 15th, because this is just how some clinical data collection protocols work. This would manifest as the death happening before the last follow-up appointment. And I have this, it's this timeline here. Let's say the index date was the 1st of the month, the death date was the 13th of the month, and the last follow-up, which um, was only known to happen in October and definitely happened before death, which is represented with the dotted line, that would be calculated to have happened on the 15th because we don't know exactly when that happened. Um, in this case, what you would see in the actual data is the days to death as 13, as in 13 days after the index date, which is a lot of times diagnosis. And the days to follow up would have a value of 15. So in this case, it might look unusual that a follow-up happened after the death date, but this is just an artifact of how some clinical data is collected. So some of these files do have metadata that states what the precision is for each time point. So you may see within an XML, for example, precision equals day. Um, so in the previous case, the days to death would have precision equals day, and the days to follow would have a precision equals month value. The other examples that the examples that I gave you, there are a couple of the most common questions, but there are many interesting things that can be found within clinical data. So if you find anything that looks out of place or weird, feel free to contact the GDC help desk. We may know the answer, or we may need to do additional research, or we may point you to a different group of people who will know much better than us. But we should be able to guide you in the right direction. So the next question is that some files have the same file name. This is rare, but it sometimes does happen in the GDC. Um, and this is because some GDC pipelines just generate files with the same names, but they will still have distinct UUIDs. So when you're downloading data from GDC, always remember that the UUIDs will be distinct. If you download the files using the GDC data transfer tool, it will put the files into folders, and each of these folders is named after the file's UUID. So no duplication or overwrite will happen. But if you're downloading from using the API, be careful, because if you're putting all files into the same directory for certain file systems, and they have the same name, they may overwrite each other, and you don't want to come back to a download with one file in your directory after leaving the lab for many hours. So be careful when doing that. Next question. Why do some math files have a header but no variants? This is an open access math here that came from the portal, and you can see that we have the header, which are a bunch of things that start with a pound sign, and then the column headers are there, and there is nothing underneath them. Users do come across this all the time, and the reason this exists is because during the variant masking and aggregation pipeline, so the ones that are used to make open access maps, variants are removed based on certain criteria. You can watch the DNA-seq webinar to learn about those certain criteria, but usually it's based on what kind of risk they are to be germline variants, which we don't want to release open access. These filters are conservative so that nothing accidentally makes its way through. And one thing about that is that that may cause all variants from a specific set to be removed. The reason they're kept available on the portal is to verify that the maps were not suppressed for other reasons. So to make sure you know that the maps weren't, um, you know, redacted or even forgotten, we want to make sure that, it's positively there, and we want to show you that there are no variants. A tip that that I've seen work is that if you're looking through like a project's maps and you want to know, and you come across a couple that have no variants, you can sort the files by file size. The empty maps should be the smallest ones, and they should have a very similar file size because header, the column headers will be the same size, and the headers above that will be a very similar size. So if you sort by that, you'll see they fall within a few bytes of each other. Um, Now let's say you know that you do need variants from certain aliquots, and you find empty maps. So you will need to access the controlled access maps, and in order to do that, you will need to apply for dbGaP access to the specific project, which we'll get into some light details about later. So the next question, how is validation performed on GDC data? Lots of GDC data comes in from submitters. Lots of GDC data comes from bioinformatics pipelines. How do we make sure this is okay? And um, we have a set of processes that we implement to ensure data quality. So the basic ones here are first the data dictionary. Any data that comes into GDC goes past the data dictionary. You can see what the schema is, but the purpose of this is to only allow certain fields and certain values and data types for these fields into GDC. So certain information can only be integers, certain information can only be a list of enumerated values. We have very few anything that is open string based. Usually those will have to be run by the help desk, but we make sure that all data coming in is something that can be useful and can be analyzed. The other um, the other process that we have is called pre-harmonization QC. These are slightly more advanced tests that go across many different entities to verify the conditions are ready for harmonization. So, for example, if somebody were to submit, a two FAQs that are paired end, but both of them have an R1 label, this test would catch that and say these are not ready for harmonization. There should be an R1 and an R2. Lastly, which I'm going to talk about for a second, but there is a lot to this. Pre-release QC and the GDC team, it's actually a few members of a few GDC teams, verifies a series of conditions before new data sets are released. So we have an idea for what's coming out for a specific data release, and we want to make sure that all the data is there, the data there is accurate, and that nothing that shouldn't be there is there. So there are many steps to that, and for information on all of these, that goes quite a bit in depth, I'd recommend checking out the GDC Data Quality webinar that happened in November 2020. So the next section is submitting data, and I'll admit this section's a little smaller because we know we don't have there's a high ratio of downloaders to submitters. But if you have any questions about submitting, feel free to contact the help desk. So this should just be two questions. So the number one, how do I submit data into the GDC? All submissions start with a data submission request form that's sent to the GDC help desk. This form is used to gather important data about each project, like including which experimental strategies, how many patients, and then the project is approved or rejected by our review committee. And this is based on the amount of data and how much the data fits in with the GDC's overall mission. So it does usually need to be a cancer molecular data. So after approval, um, the project is created at the GDC, and the clinical biospecimen and molecular metadata can be submitted through the submission portal or API, usually depending on what the size of the data is. And then molecular files are submitted using the GDC data transfer tool. So the next question is probably more of interest to regular downloaders. What kind of molecular files are uploaded to GDC? Are they FASTQ or BAM? And the answer is that submitters are allowed to submit either of these formats because, as you can see in this diagram here, um, FASTQs are aligned directly, whereas aligned reads, BAM files, are then just converted back to FASTQs and then aligned using the same exact pipeline. So it's just an extra step. The GDC doesn't mind using it. If submitting one file is easier than submitting many files, that's totally fine. So in terms of other file types that can be uploaded, we accept IDATs for arrays. For metadata, TSVs, XMLs, Excel files, and then SVS, JPEG, JPEG 2000 files for slide images. We accept many, many, many different types of data. So look in our data dictionary. If you're interested, it'll be under data format. And if you're a prospective submitter and you have like files that don't match any of these, feel free to reach out to us. We do dictionary changes sometimes for data sets that we think are important. So it's always worth asking. So the next largest section is on accessing data. So this question, how do I filter data by custom fields, is one of the most common ones we get at the help desk. And it's not always even that question because users may not know that custom fields exist. So when you go on the portal, you'll see on the left all of the different fields there. Those are the default ones. But as you know from looking at the GDC data dictionary and all of the metadata in the files, there are lots of criteria. And all of these criteria can be filtered as long as they're populated. So the way that one can get to this is by going to either add a file filter or add a case biospecimen filter. This will be on the top left of the repository page of the GDC data portal. And once you click on these, a a window will pop up and will allow you to type a specific field into it. Um, a very common one that users look for is sample type. Like before, this will tell you whether something is tumor or normal or perhaps something else. And whatever matches to that will come up. I always recommend if you're in doubt and it comes up multiple times, choose the shortest one. So in this case, I would recommend choosing sample type and not sample type ID. Um, and then once you click on this, you will see the new filter appear in the left panel. So the next question, for accessing data, how do I know which dbGaP project to apply to? Um, so we're going to get into how to access controlled data very briefly. But in general, in order to access controlled data, you need to have access to a specific project or program. Different project in the GDC, some projects are controlled on the program level. So for example, TCGA, once you have access to TCGA, you have access to all 33 projects. Other projects are accessed on the project level. So for example, I believe CPTech 3, I'm sorry, don't quote me on that one. There are certain things like HCMI CMDC. That is one that has a project level dbGaP ID. So the way you can determine this is by going to the project page in the data portal. clicking on whichever project you are interested in, and then you will see a dbGaP study accession number there. This is the one that you need to apply for. If you click on this, this is a link. This will lead you to the dbGaP page for that project. Um, so, um, and that'll give you more information about applying for it, such as, what their data use limitations are and, um, all of the interfaces for applying for data are on that website. So let's say you have access to projects and you want to see which ones you have access to. Um, you can do this by logging into the GDC data portal by clicking login. An NIH login screen pops up and you type in your credentials. Once you're done with that, your login will be replaced with your name. You will click on that and you will click on user profile, and that will show you a kind of like a TSV looking thing with some check marks. What you are interested in, and what most people will experience, is the member column. Whichever projects you have a check mark in the member column, those are the ones you have download access to for controlled access data. Um, and one correction, when I say that your name pops up, it's actually your ERA Commons ID. I know that usually corresponds with names, but just want to make a correction there. So very basic question. How can I download data from GDC? And we go into this very much in depth in the GDC Data Download and Upload Strategies webinar for November 2021. And that's where I took this diagram from. But in general, there's three options. There's the data portal, there's the data transfer tool, and then there's the GDC API. So if you have less than 5 gigabytes and less than 10,000 files, we recommend downloading from the GDC data portal. The other ones work too, but I know the GDC data portal can be pretty an easy way to access these files. If you have more than 5 gigabytes or more than 10,000 files, we recommend using the GDC API or the GDC data transfer tool, depending on what your system can do and what you're familiar with. So both of those should work. The data transfer tool does some nice features, but I understand that sometimes installing software on a work computer can be a hassle. So API is always there for you. So speaking of the data transfer tool, what do I do if the data transfer tool isn't working well? Maybe it's crashing, maybe it's downloading very slow, and the thing to do first is to make sure you have the most up-to-date version. If you go to gdc.cancer.gov, access data, and GDC data transfer tool, you will see what the most up-to-date version is, and you can then compare this against the GDC client by going to your command line, running the GDC client, and typing -- version, and that will give you the version. In this specific case, this is not the latest version. 1.6.1 is. So this person should go to the website and download the most up-to-date version. If now you're all up to date and maybe you're still experiencing the same problems, these problems can be separated into two distinct types, and by types I mean causes. Number one, there are issues with the DTT, the data transfer tool, and how it interacts with your system. It's a piece of software, and sometimes certain systems have nuances, security settings, etc., that don't play well with the data transfer tool. The other issues can be how your computer connects to the GDC network. Perhaps there are other security settings that aren't working so well there. So a good way to test this is to download a larger file directly from the GDC API. If you're having trouble with the data transfer tool and it works fine in the API, usually a data transfer tool problem. If the API isn't working, that usually means that it's a connection issue because the DTT uses the API. So if it doesn't work with the API, it won't work with the DTT either. Um, so, and if you do find that that both of these aren't working, the help desk is happy to help. Um, but a lot of times, if it's security settings, we do recommend contacting your local IT team, and they can usually get things working. But we're happy to get the GDC API working for large scale downloads if that's what you're looking for. Okay, so can I use open access GDC data for a manuscript, a for-profit endeavor, or for education? And usually the answer is yes, but there are some um, rules for accessing open access data that the GDC has, and these come directly from the NIH Genomic Data Sharing Policy. Um, these two, I won't read these verbatim, but the two major rules are don't attempt to identify any individual human participant um, from the data, and acknowledge in all of your work that this data came from the GDC and specifically came from whichever project you're downloading from. Um, if you have any doubt about what you're using the GDC data for and whether it's ethical, legal, etc., we recommend contacting the NCI Office of Data Sharing. Um, we contact them all the time for either internal questions or questions from users, and they will usually know the exact right answer. So they are experts at that. We recommend contacting them for um, any particular situation you have any doubts for. So one question you may have is how to access data from TCGA marker or other landmark cancer genomics paper. Um, you may read a manuscript and it will say data can be found in the GDC. And some of the data is just on the portal. You go to the specific project and download it there. But some data is supplementary and may not fit in the GDC data model, like a table of samples, for example. GDC cannot process that. So these are stored on GDC publication pages. You can go to gdc.cancer.gov, about the data, and then publications. About the data is like a tab. So these contain links to these resources and files that aren't exactly what GDC processes. So and there are some control files in there, and these do require dbGaP access depending on the project. So it requires dbGaP access to a specific project. It's not like, publication pages are all the same project. So if it's a TCGA project, you need access to TCGA. If it's a Target project, you need access to Target. And then just to show you kind of what one of these pages looks like, there's a lot more to it, but this is like kind of what the download looks like. The open access data can be downloaded either via manifest or can be just clicked on directly using these links that'll download them directly to your browser. But if you're downloading controlled access data, these need to be run through the data transfer tool or API. So you would need to download the manifest and use that to download the files. But something that I don't show here is that the controlled access files are described in the page. So you will see like whatever brief description the researchers shared there. Okay, so something I've been referencing a lot during this presentation, how do we get access to controlled data? And this is a very brief explanation, but the general flow is that you get an ERA Commons ID, you use that ERA Commons ID to go to dbGaP, and at dbGaP you can apply for a specific project. Each project, like I've mentioned before, requires a PHS ID. For example, TCGA is 000178. We go into this very in-depth in the January 2022 webinar about obtaining access to control data in GDC. And during that, we show a video that dbGaP makes directly on how to get access to a specific project through the dbGaP interface. So if you're looking to do that, our video or the dbGaP video are a good place to start. Next question, real quick. Which units are used for file size in the API? Answer is bytes. If you use the API, you'll see a number there that is in bytes. The bytes values are then parsed out to display more larger units in the data portal. So this, for example, this is 135.366, etc. Gets parsed out to be 135.37 gigabytes. If you do need to verify an exact size, we recommend using the API, and here is a curl command through the command line that can be used to get a file size. So it's basically API URL, files, and then the UUID, and this will automatically give you the file size in your response. So why do some projects have whole genome sequencing data but no structural variant files? And this is an example of a question a user might have, but in general, it works like this. GDC pipelines are run in stages, and they can be released in stages. So depending on what the submitters want or are authorized to release, they may have alignments, which then undergo variant calling, which are then annotated to be math files. So in some cases, the data owners may say, we're allowed to release the alignments, but the variant calls are being processed internally, perhaps for a manifest or an analysis working group, and they are not allowed to be released yet. So you may go on to the portal, find that a project you're looking at has a specific experimental strategy, like whole genome sequencing, and only find alignments there. And that is usually why. In some cases, it's that we are only authorized to release a subset of files until either a publication is released, in that case they'll inform us, or a specific date is reached. In other cases, the pipelines that generate the missing files may not have existed when the project's data came to GDC. So what we try to do is run the new pipelines later and release them during later data releases to restore a project's completeness. So in data release 34, you may have seen that a lot of projects that came into the GDC a bit earlier now have transcript fusion data. And that is because the transcript fusion pipeline didn't exist when these projects came into GDC. So the last section is the GenCode updates and other bioinformatics topics. This is a bit shorter, and apologies, it's pretty stormy here, so you may hear some thunder. So which reference genome is used at the GDC? So you've probably heard the GDC aligns to the GRCH38 reference genome, but there are multiple versions of this. And the one that's used exactly is D1.VD1. If you're interested in the FASTA format of this file, you can go to the GDC reference files page by going to gdc.cancer.gov about the data and GDC reference files. And you'll see that you have the version of the reference genome followed by .fa, that's a FASTA file, and then it's a tarball. It's like a little less than a gigabyte. And then some more data, for example, the decoys and the viral sequences can also be downloaded from that page. So another question we get is how often the reference genome is updated at GDC. And the answer is right now that GDC has not updated the reference genome, as there haven't been any updates to the current reference genome that were deemed significant enough to change. And you may ask, who makes that decision? And it is a management decision, but in general, changing the reference genome requires replacing nearly all GDC files, and that involves rerunning all of the pipelines, organizing them, and releasing them all back to the portal. So the reference genome will need significant changes before it will be replaced at GDC. However, one change the GDC has made is the gene model. So when GDC first started, the GenCode v22 version was used, and now v36 was used as of data release 32. This involved replacing any file that was affected by the gene model. I know we're running a little short in time, but where are the copy number variants on the exploration page? So before data release 32, which is the GenCode upgrade, the exploration page featured TCGA copy number variants that were produced from GISTIC. So after this data release, the GISTIC files were deprecated, and now those same variants were produced but using the ASCAP pipeline. Note that the raw GISTIC files are still available for download. If you have the UUIDs, you can, and I'll show you how to get the manifest for that later. So the GDC underwent a review for how ASCAP data should be visualized, and the data should be returning to the Exploration Portal in the next 2 data releases. So if you're interested, all the files are available. Just right now, as of August 2022, the exploration copy number variants are not available. So why did GDC remove HTSeq for gene expression quantification? As of data release 32, HTSeq workflow was removed from GDC. HTSeq was the original software used by GDC for gene expression quantification back in 2016. Since the GDC's launch, we've adopted the star counts pipeline, which is faster and provides additional stranded counts, whereas HTSeq only provided unstranded. I think there's ways to set it to do stranded, but star counts was much easier. The GDC, to make up for the missing FPKM and FPKM upper quartile normalization, has added these columns to the star counts file so that they are present. If you are looking to download HTSeq files from before DR32, this definitely can be done. Although they aren't in the portal, they are still downloadable, and this applies to any pre-data release 32 file except maybe the BAM files, depending on which point. I think there's about half a month left to download those, in which they'll be guaranteed to be there. If you do have a previous manifest with HTSeq files, that should still work with the GDC data transfer tool. And if you don't have a manifest, don't worry. We do have manifests from pre-DR32 releases available in the GDC documentation site in the data release notes. So you click on data and you click on release notes. I recommend going to data release 31, as that's the most complete data set before the GenCode upgrade. And then you can filter them with Bash or with a spreadsheet program like Excel. And then you can just pass that manifest directly to the data transfer tool. You will get HTSeq files. So those are still available. So before I get into the review quiz, are there any questions in our Q&A panel? Let me see here.
Hi, Bill. So far, I don't see any questions.
Okay. Sorry, it took me a second to find the Q&A button, but here we go. Okay, so we have 10 minutes left for each of the review questions. I'll give everyone 10 seconds, and if you'd like to participate, feel free to type your multiple choice answer into the chat box. So starting out, easy one, true or false, users need to first sign in before they can see which projects they have controlled access to in GDC. All right, the answer is true. In order to see the controlled which projects you have access to, it does need your sign-in information, and that's how it's determined. Next question. If two files have the same file name, they should be identical files, true or false. Yes, I saw some falses come in. That is correct. Two distinct files can have the same file name, but will always have unique UUIDs. Although with most files in GDC, they will often have unique file names. All right, next question. I filtered a set of files down to 5,000 files. I need to filter it more, but I cannot find filters for the attributes I need. What should I do? A, download the entire file set and write a script to determine whether or not the file matches your criteria. B, use the add a file filter function on the GDC data portal. C, hire an intern to manually investigate each file and present the filter list to you at the end of the day. Or D, use the auto find function of the GDC API. So the answer is B. There's an add a file filter function on the GDC data portal. I do want to mention that D is a fake feature that does not exist, but I do wish it did exist. Next question. I'd like to download 100 gigabytes of files. How should I accomplish this? A, the GDC portal, B, the GDC API, or C, the GDC data transfer tool? Both B or C would be correct depending on your situation. And yeah, like I said before, if one doesn't work for your particular system, always feel free to try the other. Okay, where can I download a manifest that contained files in the GDC that aren't on the GDC data portal? The GDC data release notes at docs.gdc.cancer.gov, the GDC data portal at portal.gdc.cancer.gov, a secret AWS bucket, or the GDC API at the manifest/old endpoint. All right, the answer is the GDC data release notes at the doc site. And I do want to mention that the GDC API manifest old endpoint isn't actually real. So don't try to use that, you'll just get an error. That is the end of the quiz. Thank you all for joining and for participating. And if anyone has any questions, I think we have about 5 more minutes and I'll be happy to answer them. All right.
Hi, Bill. I think we have a question there.
All right. Is there a way to extract mutation data with expression data for a project? Yes, there is. So I can actually demo this real quick. So if you were to go to the GDC data portal, and let's say you wanted to know which cases did have expression data. What you can do and I think you specify TCGA, so let's filter by TCGA right here, and then in the files tab here, you could filter for those with expression data, and I'll use RNA-seq for this one right here. So these are the cases that have expression data. There are 10,261, so it's just a little bit too big to make a set, but that's okay because what you can do is go over to the this button here, view 10,261 cases in exploration. If you click on that, this might take a little bit because it is querying 10,000 cases, but if you go to the exploration page, now it is filtered down to these 10,261 cases, and you can do whatever you want with this mutation data, which would be, um, 19,000 different genes with 2.1 million mutations associated with them. So, for example, you can look at the survival curve, and this is filtered down to those specific cases.
Okay, does anybody have any additional questions for Bill?
All right. Well, thank you all so much for joining and for participating in the quiz at the end. I really do appreciate that. And yeah, I hope you all have a good rest of your day. Thanks, everyone.
Thanks, everybody.